My name is Kara Brook, and I'm a conservation biologist and disease ecologist who studies zoonotic infections of fruit bats in Madagascar. I first started working in Madagascar in 2010 as a sustainable development intern for World Wildlife Fund. They say that those who drink the water of the Manangareza River never stop coming back to Madagascar.
Over 60% of emerging human infections are zoonotic, meaning derived from an animal reservoir. Bats host more zoonotic viruses per capita than any other mammalian order, but appear not to experience ostensible morbidity or mortality upon infection with viruses such as Ebola, Marburg, Hendra, Nipah, and SARS, which cause extreme pathology in other mammals. As the only volant or flying mammal, bats are unique in many ways. Evidence suggests that novel molecular pathways developed to enable flight, a highly metabolically demanding activity, may have had cascading consequences on bat longevity and antiviral immunity. At the within-host scale, my research employs a combination of tissue culture and modeling tools to understand these exceptional molecular pathways, including constitutively expressed antiviral defenses, which facilitate bat tolerance of otherwise virulent pathogens. At the population level, along with the help of the inimitable Equipa Fanihi, Malagasy for Team Fruit Bat, I track longitudinal viral and immunodynamics in flying foxes in Madagascar. Our hardworking team of Malagasy postdocs and PhD students, paired with American research technicians, works full-time in the field and the lab, capturing bats every month to collect a suite of biological samples, which we assay using serological and PCR-based tools to investigate past history of exposure and current infection status. We work in Madagascar, the 8th continent, where Asian and African lineages of Old World fruit bat and their concomitant viruses coexist in a closed island ecosystem. Across Madagascar, fruit bats are hunted widely as bushmeat, offering ample opportunity for zoonotic transmission. A subset of captured fruit bats undergo anesthesia to allow for tooth extraction. These bats set down annual layers of a tissue called cementin, and by slicing, staining, and counting the layers, we can learn the fruit bat's age, much like counting the rings in a fallen tree. We use these age data to bolster our modeling inference for population dynamics, where our estimates of adult survival rate for Pteropus rufus, the largest and most heavily hunted Malagasy fruit bat, suggest that this species is in serious population decline. On the infection side, compartmental transmission models fit to age-structured serological data are converging on a hypothesis by which bats host their viruses persistently across the year, but shed them only during periods of nutritional or reproductive stress. I am dedicated to research, outreach, and education in Madagascar for the rest of my life. In addition to Equipa Fanihi, I am the co-founder and co-organizer of E2M2, Ecological and Epidemiological Modeling in Madagascar, an annual course that teaches statistical and mathematical modeling techniques to Malagasy and, more recently, to a subset of foreign students. This year, in conjunction with E2M2 and my colleague Elizabeth Toomey, I also helped organize the first ever Women's March Madagascar. To me, events like the Women's March acknowledge that the world as it is a very unfair place, and affirm our commitment to trying to change that. I close with the words of a wise U.S. Senator and one-time Secretary of State in Malagasy and in English. Atau awavi aonsaina famatanzaka, simisi lanza aritain ni lananario, arikua mendrika arimananzu ni hahazu izaizavatsu si pahapahana misi amfiainanario. Never doubt that you are valuable and powerful and deserving of every chance and opportunity in the world.
Samanofiano Isakale Tidingi kweki Tidinge Atungavano Ankila kwa ketu Ipetano Ankila kwa ketu Ibudano Anila kwa keo Sentangi nyako